Hi guys, this is Liang, Liz and Valerie's team from the biomedical program. The following video will describe how the lab processes a breast tissue biopsy before handing it to the pathologist for examination. After a biopsy, a breast tissue then is sent to the lab for pathology examination. In case of malignancies, further molecular and proteomics tests will be performed. Once the breast biopsy has been collected, the medical staff puts it in a container and in a biohazard bag with the corresponding requisition and delivers it to the lab. Once it is received in the lab, the clerks must ensure that the correct specimen was delivered with the proper requisition. If everything matches, the specimen is scanned and marked as received in the lab. After a specimen has been identified, the next step is fixation. In this step, the cassette is put into a fixative for 12 to 24 hours. This is done in order to keep the tissue the closest as it was in the body in order to get accurate results. The most common used fixative is 10% neutral buffered formalin. Once fixation is done, the next step is processing. So after the 24 hours have passed, the tissue is loaded into a processor and the processor cycle is started. The tissue will be submerged in ethanol to remove all water from the tissue. The ethanol is then removed from the tissue with a clearing agent. The last step during processing is called wax impregnation. This involves the controlled removal of clearing agent from the tissue and replacing it with molten wax. The processing cycle can last up to 12 hours. Once the tissue has been infiltrated by the wax, the specimen is now ready for embedding. To start the embedding process, the tissue is taken out of the processor and brought to the embedding machine. The lab tech will open the cassette, make sure there is no tissue stuck on the lid, and then discard the lid. They then choose a mold of the appropriate size and transfer the tissue with forceps into the mold, ensuring proper orientation. The mold is then filled with wax and put onto a cold plate while holding down the tissue to prevent floating. The bottom of the cassette is then put on top of the mold and filled completely with wax. The now embedded tissue is left off a cold plate until the wax has fully solidified. Embedding is done to allow the cutting of very thin sections of tissue which would not otherwise be possible. After embedding, it is now time to cut the section for the slides. The block is loaded onto the microtome. The lab tech will first coarse trim the block to remove the excess wax and expose the tissue. The section thickness is then reduced to 4 micrometers for the cutting of the tissue section for the slides. The lab tech then proceeds to cut the desired amount of section and drag them onto the warm water bath. A charged slide is then used to pick up the tissue from the water bath. The charge of the slide will cause the tissue to stick to it. Once all the slides have been cut, the slides are put into a dryer oven to allow the slides to completely dry before staining. For staining, the slides are loaded onto an automatic stainer where they will be stained in order to visualize the different components of the tissue. The slides are then given to the pathologist for examination. Along with the biopsy specimens sent to the pathology lab, sometimes other specimens are sent to other labs like the chemistry lab in this picture, where the patient's blood is sent and then it is analyzed for tumor markers or other tests that the doctor might have requested.